Hello and welcome to NEO. NEO is a building at the Karolinska Institute in Sweden. This is where we are, and it's a very colorful building. And now we're going to go to the live cell imaging facility, which is obviously located on the 7th floor. So here we go, and this is the door to the lab. Okay, let's go. So normally this facility is very dark, but I have turned on all the lights for you. So you can see that there are 8 compartments, and in each compartment there is a microscope. We are Nikon Center of Excellence, so we have a lot of Nikon microscopes, but not only, we also have other microscopes. So all the Nikon microscopes look a little bit the same, but they're actually all different. So Pegasus, for example, is a single point confocal with a resonance scanner. And you can see how we have organized our microscope with, we try to put most of the equipment above the microscope to save space. So this is Orion. This system is a single point confocal with a spectral detector, and it also has wide field turf and storm super resolution on the right side. Now we move on to the next microscope, which is called Twitty. Twitty is our super duper machine. This is the latest beast. We have a single point confocal on one side, and on the other side we have a spinning disk / wide field with 2 different CMOS back illuminated CMOS cameras. We're very happy with them. And at the back of Twitty we also have Alveol from Primo, and this is a micro patterning device. Really cool machine. Now let's look at Victor across the corridor. Victor is a wide field on one side and a spinning disk on the other side. There is someone imaging, so I don't want to disturb too much. It also has an incubator, as you can see. All our microscopes have large incubators, and that allows our users to come in and out with either fixed or live samples and without thinking too much about it. So we have a lot of live cell imaging in here. Now we move on to Dragon, just next door, and Dragon is also a single point confocal with a resonance scanner and a spectral detector. And this is Zeus. It's an upright single point confocal from Zeiss with a multiphoton laser and a gas detector. The second non-Nikon microscope in this room is Aurora. Aurora is from M2 Lasers. It's a light sheet microscope, so you can see the 2 objectives and the large incubator. We can image on any type of imaging medium with Aurora, and we are super happy with it. We get a very big field of view with a better than 1 micron resolution across the whole field of view. This is really great. So that's a very nice machine. Here is Aurora. Finally, the last microscope that we have at the LCI is Minerva, and Minerva is also a Nikon microscope with an MMI laser capture microdissection and also a cell picker device, and we also use it as a slide scanner. Okay, so now we have seen that at the Nikon Center of Excellence side of our imaging facility, we have 4 single point confocals and 2 spinning disk confocals. We also have 4 wide field systems. All our systems are equipped with high throughput, high content screening modules. We also have a super resolution storm microscope. And on all our systems, we also have a feedback microscopy, which means that we can do complex experiments where we have, for example, 1 round of imaging, 1 round of image analysis, and then another round of imaging based on the result of the first image analysis. Then we also have artificial intelligence, and additionally we have a laser microdissection and a micro patterning possibility. Outside of the Nikon Center of Excellence, we have light sheet from M2 Lasers, and we also have a Zeiss multiphoton microscope. We can image fixed and live samples from very big to very small, like cells, tissues, organoids, zebrafish, whatever. We provide expert help in sample preparation and experimental design, as well as image analysis. We have a virtual server and software on the server, as well as an in-house image analyst who will help you make your pipeline. Our facility has a strong emphasis on education, and therefore we have an extensive tailor-made one-to-one training, as well as a yearly microscopy course. So you're welcome to visit our website or to contact us. That's it. I hope that you enjoyed the virtual tour of the live cell imaging facility, Nikon Center of Excellence at the Karolinska Institute in Sweden. Bye-bye.